Okay students, so let us talk about the different kinds of structures that we encounter and that we observe in a particular cell, starting with the outer layer, that is cell wall. Okay, as you can see, the cell wall is the most prominent part of plant cell structure. It gives plant a body, a shape that you may encounter of different types. Okay, you may find it hexagonally placed, as in case of many kinds of cells, as you may have prepared onion peel slides, or you may find it irregular, wavy, whatsoever. All of these particular structures and shapes are being designated with the help of cell wall. It is made up of cellulose, hemicellulose and pectin. That is really important to remember. Okay. Other than that, it protects the plasma membrane and other cellular components that are essential for cells functioning, that we'll discuss as we move particularly in through. Alright. So, if I talk about the structure of cell wall, it is mainly tri-layered or three-layered. Okay. There is middle lamella, then primary cell wall and secondary cell wall. Okay. So, what is the function of a particular cell wall? A cell wall provides a framework and definitive shape to the cell, as I discussed with you. Alright, it gives rigidity and support to the tissues. Okay, like our bones that supports our body and internal organ structure and organization, the cell wall also gives the rigidity and support. Okay, cell wall gives mechanical strength to facilitate the plant and supports its long and tall body. Okay, you may know that plant grows from a little sapling and it takes lots and lots of divisions, lots and lots of cellular and metabolic growth in order to achieve that girth, that strength that plant has, and that is ensured with the help of cell wall. Okay, it helps in balancing and maintaining osmotic pressure and prevents bursting of cells. As we have spoke about the transport system about the cell in the previous parts of this particular chapter, we know that selectively permeable membrane, selectively pressurized sensitive membrane is present in order to get this particular cell surviving and get this particular other structures not invading the cell. So cell wall provides protection to that cell. Okay, it helps in transport of water and minerals over the long distances. That in the higher class you may learn about the pathways through which water particularly flows. It may form channels between the cells or it may form particular ways in the gaps between the cells. Okay, so let's leave it there for the higher classes. We should move on to the next part. The next part that we are going to talk about is nucleus. The nucleus was discovered by Robert Brown in 1831. The nucleus is a double membrane bound dense protoplasmic body which contains the genetic information or the genetic codes for which any cell, for which any organelle codes for. Okay, it ensures all the cellular metabolism and encloses the genetic information of the cell, which is needed for carrying out, which is needed for multiplication, which is needed for every growth prospect and aspect of the cell, eventually the body as well. Okay, the nucleus is considered as the controller or director of the cell. Okay, whatever is happening in the cell, you may call it functioning, you may call it any kind of metabolic process, this all goes through with the help of nucleus. That nucleus directs this particular organelle has to do this or that. Okay, so if I talk about the structure of nucleus, nucleus have firstly the nuclear membrane. Okay, it is double layered, porous, made up of lipids and proteins or lipoproteins as in certain, and it separates the nucleus from cytoplasm. All right, it is transparent, dense and ground substance of nucleus which is present on the outer periphery or the surface particular area. Okay, if I talk about the second part, that is nucleoplasm. Nucleo means nucleus, plasm means fluid, that is nucleus fluid. Okay, so there are chromatin fibers and nucleoli that are mainly suspended in it like a wavy pattern arrangement, and it is surrounded by a nuclear membrane. Okay, it is slightly viscousy or slightly flowy if I talk about the in comparison with nuclear membrane. Okay, The third part is nucleolus, which is spherical, non-membranous and vary in number. It is mainly rich in proteins, okay, and RNA molecules. It is the particular site for the formation of ribosomes. We'll talk about ribosomes in the later part, okay. Other than that, we have chromatin material, which is a thread-like spindle coiled structure which contains our genetic hereditary material or the DNA in cases, okay, DNA or RNA, whatever it could be at the case. So what is the function of nucleus? It is the main controlling center of the cell where everything gets controlled, regulated, channelized, you name it. Everything is done through nucleus. Other than that, it has a major role in cell division. Along with it, it regulates metabolic activities that are mainly controlled by the enzymes. Alright, I hope this part has been clear with you. Now we talk about centrosomes chromosomes. We talk about chromosomes now. So chromosomes are thread-like structures in which DNA is tightly packaged within the nucleus. Okay, chromosomes are the main particular structure which contains the genetic material in a pattern way arrangement or fashion. Okay, DNA molecules contain the information that is necessary for performing any kind of metabolic and cellular activity. Your height, your growth, your hair color, you name it, every part of it has been carried forward from your previous parental generation in yourself, whomsoever is watching this video right now. And you know, obviously the superior characters, the expressive characters are being present in yourself. The recessive characters might be, but they are not expressing enough. All right, and this all is being done with the help of these chromosomes, which carry all those genetic information and select the superior quality that has to be present. In order, we can say we are the best product of our own parents. All right. So now I talk about cytoplasm with you. The cytoplasm is a living colorless semi-liquid which is homogeneous in nature. Okay, it occupies the major part of cells and it is consistently moving. It is not static, it is dynamic. Okay, now we talk about the main organelle or the powerhouse of the cell that is known as mitochondria. Okay, mitochondria is a double membrane bound organelle. As you can see over here, there are different kinds of layers, particles. It has its own DNA. Okay, it has its double membranous structure along with matrix and proteins, that is ribosomes. Okay, so mitochondria is also known as powerhouse of the cell, and mitochondria synthesize respiratory enzymes which help in generation of energy in the form of ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate. Okay, 3 phosphate molecules are being attached over here, and in our body, you name any kind of energy expenditure process or reaction wherever is required, this ATP has to be there for the same. Okay, so that energy can be given, energy can be transferred like a currency. Okay, like if you want to purchase something, you pay in a particular currency amount, like pay some rupees, right? Similarly, if you want to get some work done, energy has to be required, and energy synthesis is done at mitochondria. Okay, okay, so now we talk about the next cell organelle, that is endoplasmic reticulum. Endoplasmic reticulum or ER is a large network of membrane bound sheets. Okay, those wavy kind of sheets that we have seen in mitochondria, that is humongous over here. Okay, there are 2 kinds of endoplasmic reticulum that we see. There is rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, so Why rough endoplasmic reticulum is known to be rough? Because it contains ribosomes at its layers or at its surfaces, whereas smooth endoplasmic reticulum doesn't have that, right? So structurally that is seen as in a difference, okay? Now I talk about the next structure, that is ribosomes. Ribosomes are small nucleoprotein particles that we have been seeing or watching about, or you may be observing it in mitochondria and now in endoplasmic reticulum. So what is ribosomes in general? Ribosomes, small nuclear protein particles found attached to rough endoplasmic reticulum and free form in the cytoplasm of the cell. Okay, so we have seen it at three ways right now. That is cytoplasm, that is mitochondria, and endoplasmic reticulum. Okay, each ribosome is made up of two subunits. One is a smaller subunit and a larger subunit. Okay, they provide site for synthesis of proteins and hence are called protein factories. They basically prepare that particular site where protein formation can occur. Mitochondria makes its own protein. Okay, cytoplasm, you have these protein units, and endoplasmic reticulum's main function, or rough endoplasmic reticulum's main function, is to synthesize proteins. Okay, so these are called as protein factories of the cell. Something extra that I would like to talk to you about, what are these subunits? These subunits are named after a scientist known as Svedberg. And these units are known as Svedberg units. Okay. So now there is Golgi apparatus. It was discovered in 1898 by the scientist Camillo Golgi. Hence it is known as Golgi's apparatus. Golgi complex has a convex forming face or cis face. Okay, you will encounter this cis and trans arrangement in the later classes or higher classes, but to know just now, it is the convex forming phase and concave maturing phase. That means in the forming phase, it is like a convex shape, and in the later mature stage, it is concave. Okay. Forming phase receives vesicles from ER or endoplasmic reticulum. That mature phase secretes vesicles. Okay. These are known as dictyosomes in a plant cell. So in a plant cell, you won't say that they are vesicles or Golgi apparatus. In plant cells, they are dictyosomes. Okay, very important to note that. And it transports synthesized substances from cell to the outside. Okay. Now I talk about lysosomes. Lysosomes are spherical bags-like structure, like typical polybag kind of a structure you may imagine. When a cell gets damaged, lysosomes play into the part. They burst and the enzymes digest its own cell. That is why they are known as suicide bags or suicidal bags of a cell. Okay, because if they rupture, if they burst, they may damage the complete assembly of the cell. Okay. So now we talk about centrosomes and centriole. A centrosome is a cellular structure that involves the process of cellular divisions. You may know about mitosis and meiosis in earlier classes. That has been done with the help of centrosomes and centrioles. Okay, each centrosome contains paired barrel-shaped organelles. Okay, and all centrioles made up of protein strands are known as microtubules. What happens during the gametogenesis, during the splitting of the gametes or the chromosome that we spoke about, these splitting has to be carried at the particular stage where the division has to occur. And for that carrying purpose or leverage purpose, these centrosome and centrioles secrete that proteinaceous fibers that hold these chromosomes in such a way that they can be segregated and they can be splitted up. Okay, I hope it has been clear with you. So centrosome initiates and regulates cellular division in animal cells, and they develop mainly spindle fibers with the help of asters, which is like star kind of a shape structure which secretes those spindle proteinaceous fibers that attaches the chromosomes. Okay. Now we talk about plastids. So plastids present only in plant cell. There are three types of plastid, which is chromoplast, leucoplast and chloroplast. Chrome means color, so these are colored plastids. Leuco means white, so these are white or colorless plastids. And chloroplast, chloro means chlorophyll, that these plastids contain the chlorophyll. Okay, and plastids like mitochondria have their own DNA. One thing that you need to remember, plastids are kitchen of a plant cell. Plastids are known as kitchen of plant cell. You may know about the process of photosynthesis, and for that to occur, to capture the light, to synthesize the food, or the glucose, plastids come up in the play, and they basically synthesize those chemical energy and sunlight energy in order to generate the food. Okay, that's why plastids are known as kitchen of a particular plant cell. Okay. Now we talk about vacuoles. Vacuoles are enclosed compartments, humongous compartments, which are water-containing inorganic and organic molecules. Dujardin coined the term vacuole, and vacuoles are single membrane bound storage sac-like structures. Okay, these membrane structures or the membrane vacuoles are known as tonoplast. Okay, these also store certain kinds of nutrition, mineral materials, etc. for the usage in future use. Okay, now we talk about peroxisomes. Peroxisomes are involved in catabolism of fatty acids. Breakdown, we may say. The crystalline core contains protein called catalase. Okay, and they are specialized in detoxification of toxic substances from the cell.